Hello, my name is Matthew Van Dam, and welcome to my talk. I'm a research scientist at the California Academy of Sciences, and today I'm going to be talking about how the Easter egg weevils got their spots. Phylogenomics reveals malaria mimicry in Pachyrhynchus. If you would like to know more about the talk that I'm presenting, you can check out these two papers that are the basis for this talk, if you like. Pachyrhynchus is a genus of weevils that consists of approximately 145 species, 93% of which are endemic to the Philippines. Pachyrhynchus has been an interest to naturalists dating back to the time of Wallace, where he noticed their smooth, hard, and brilliant colored cuticle, as well as other insects that mimic them. And here you can see these are two Doliops species, Cerambycids, trying to look just like these nice Pachyrhynchus. And recently it has been demonstrated that indeed their hard cuticle is unpalatable to predators and that their coloration acts as an aposematic defense. And here are two very nice experimental ecology papers demonstrating this. So what affects the color patterning in Pachyrhynchus? Is it the composition of the local mimicry complexes, as well as interacting with the biogeography? And as well, the genetic underpinnings, the genes regulating color patterning. To address these questions, we need a good genome to start and a phylogeny. So we generated a chromosome-scaled genome for Pachyrhynchus sulfur maculatus. And one of the interesting things that we noticed is that it appeared to have this rhabdo-like conformation where the centromeres and telomeres are clustered at opposite ends of the nucleus. And as you can see in this high-C contact heat map, each one of these boxes representing a chromosome, that there are hotter, more red patches along where you find the centromeres. So that's one way that you can read this and figure this out. The centromeres are actually in contact with each other, so that's cool. So some other interesting things about the genome is that it's relatively large and mostly composed of repetitive DNA elements. And then getting to the synteny, we wanted to look and see what sort of syntenic patterns there were across Coleoptera, what were the general patterns of synteny. And these are colored bars on the top are the Pachyrhynchus with a respective, another respective beetle that's compared against on the bottom in the gray bars. And these are the Busco genes mapped between them. And as you can see, some chromosomes tend to show a great amount of conservation. So there's very few translocation events between chromosomes. However, they're relatively shuffled along each chromosome. And here's just another way of looking at that. As you can see, they're sort of shuffled like a deck of cards along each chromosome. One final way of looking at that is we can go back to our high-C map, where we see our centromeres and go to this chromosome, chromosome 11, as that was relatively conserved amongst all of them, very few translocations. Plot our centromere, since we know what that is, and then plot the Busco genes, and you can see they really are quite shuffled. And this is a pattern unlike what you see in Diptero, where there's conservation on either side of the centromere. One final thing we wanted to look at was the syntenic patterns across all of insects. So do different orders display different decay rates of synteny? And to do that, we calculated a gene order conservation score and computed a pairwise matrix between all 143 different insect genomes at a result of chromosome scale at the time. And so how does synteny decay? Are these curves linear, exponential, or power law? And are these curves different between different insect orders? But remember, each one of these pairwise comparisons is confounded by their phylogenetic relatedness. So we'd expect things that are more closely related to each other to have more synteny, things that are more distant related to have less. So to get around this, we unfurled the matrices and then permuted the synteny matrix, holding the phylogenetic distance matrix constant to generate a null distribution, which we could then assess statistical significance for our different questions. And indeed, we did find that it's an exponential decay rate of synteny and that different insect orders do seem to have slightly different rates of synteny decay. Lepidoptera being an exceptional one, and even if we take that out, they still supports a model where there are different rates of synteny decay amongst orders. So getting back to our Pachyrhynchus phylogeny, we wanted to get a really good, robust phylogeny for it. So we wanted to maximize the number of loci. So here's Pachyrhynchus on this relative scale. So we want to try to get all of these loci. We don't want to use a probe set that's designed for things that are really distant related, where you only get a few. And so we accomplished this. We got about 10,000 loci, approximately 6 million base pairs total. And then we binned UCE loci into 25 kb bins. So we had our chromosomes from our base genome and just went down each chromosome and grouped loci that were found in 25 kb bins together into slightly longer, more informative loci, as that's been shown in our systematic biology paper, to improve gene and species tree support, and as well as give you more accurate topologies. As well as we looked at partitioning and model selection. We found there's many different partitioning schemes used, not just 3 with a flank to either side and a central core. And as you would expect, as you get longer and longer loci, they tend to maximize the support, more informative sites, and binning seemed to help as well. So here's our phylogeny. So the take homes from the phylogeny is that indeed we do see these color patterns, although they're very ornate, they do pop up independently along the phylogeny. And as that what you would expect for cases of Müllerian mimicry. We also want to look at the biogeography. So we dated the tree and then reconstructed ancestral areas. What we found is that most likely it had a Luazon origin, and then it went to separate islands where there were in situ radiations, but never back colonized Luazon, which you might expect for beetles that have mimetic patterns. And there's also many independent origins of color patterning, as you can see here, via ancestral state reconstructions, as well as we found that you go through, when you have these polymorphic species, or species with these open bands, you typically go from a solid state banding pattern to an open and filled type patterning within a species, to then just simply the open banded form. So you don't go directly from one or the others, you go through this intermediate state where you have both present in a species. Looking at some notable mimetic complexes, you can see that they're widely distributed across the phylogeny. And another thing that we noted just looking at museum specimens is that species that are most abundant tend to drive these patterns. So the ones that are most numerically abundant tend to drive the other species to look like them. With that, I'd like to thank you for your attention. I'd like to thank my co-authors, especially Athena Lamb, who did the lab work, as well as Annalyn Cabras, who did a lot of the writing and specimen acquisition, James Henderson, who helped tremendously with the computational side of things in bioinformatics, my brother Alex Van Dam, who helped with the probe design, and Andy Romager, who helped with the synteny permutation test, which was a novel technique that we developed. That, I'd like to thank you again for your attention and plug these papers one more time if you would like to know more about what I've done. Okay, thank you.